Hi everyone, we're at the top of the hour, so we're going to get started with today's live webinar. Thank you so much for joining us. My name is Christina Asa, Scientific Application Specialist with FUJIFILM Visual Sonics, and I'll be moderating today's session. The title of our talk today is In Vivo Quantitative Molecular Imaging for Receptor-Specific Contrast and Image-Guided Surgery, and our presenter is Shazia Bano from Harvard Medical School, Massachusetts General Hospital. A few housekeeping notes about today's webinar. We will be recording the webinar today, and the recording will become available after for anyone who is unable to join us today. All lines are muted for the duration of our session, so please use the Q&A button at the bottom of your Zoom panel for any questions you might have throughout the presentations, and questions will be answered at the end of our session. We do expect our presentation to be about 20 minutes, leaving us 5 to 10 minutes for questions. Dr. Shazia Bano is currently a postdoctoral research fellow at Harvard Medical School, Massachusetts General Hospital. Her doctoral thesis in the Department of Physics at the Islamia University of Bahawalpur, Pakistan, explored the biomedical applications of multifunctional nanocomposites. Her major research interests are on the interface of nanotechnology, oncology, and cancer therapeutics, with a major focus on developing complex, clinically relevant nanoplatforms. Her research goal is improving the efficacy of molecular targeted photodynamic therapy-based combination treatments for cancer. Without further ado, I'm going to pass things on to Shazia.
Hi, thank you, Christina, for a kind introduction, and good morning, everyone. And thank you for signing up for today's webinar. And hopefully you are hearing me well, and you are all safe wherever you are. Thank you for the Visual Sonics for providing me the chance to share our work with the other people. So today I will be talking a little about our work in vivo quantitative molecular imaging for receptor-specific contrast and image-guided surgery. So this work is basically addressing the challenge of complexity of accurate evaluation of targeted nanoconstruct, the engagement with the tumor receptor in vivo, and this work is building a concept of a non-invasive paired nanotracer imaging approach, which is reporting on the differential tumor kinetics of targeted nanomedicine. So as we all know that non-invasive fluorescent imaging provides real-time anatomical, functional, or molecular information about the operating field during the surgery. And as a principle, a fluorescent contrast agent is administered intravenously or topically to the subject prior to the surgery. During the surgery, the agent is visualized using a fluorescence imaging system with the adequate excitation light and a camera that is sensitive to the fluorescence emission light, and that actually captures the display for the surgeon. So the surgeon follows the fluorescence margin and outline the disease for the surgery. These fluorescent images provide real-time guidance to assist in differentiating the diseased and the normal tissues. However, they provide the weak specificity. And the presence of the microscopic residual tissues away from the bulk tumor are one of the major causes of the recurrence after the surgeries. So to this end, accurate delineation of precise tumor margin is required to achieve the complete resections and to avoid the chances of the recurrence and metastasis. So overcoming the limitations of the fluorescence-guided surgeries to enhance the selectivity, therapeutic antibodies, and especially the one which are labeled with the fluorophore that emit in the near-infrared region of the light spectrum, are favored by the surgeons as they are considered to be tumor selective and have the advantage of better depth penetration within the tissue. However, the interstitial presence of unbound and non-specifically accumulated probe because of the enhanced permeability and the retention effect, which is a well-known phenomena for the cancers, give rise to a false positive detection of the tumor tissue. and its margin. So because the uptake of a probe in a specific region of interest is not just governed by the region's targeted receptor expression, but there are other physiological factors that play a role. And the weak selectivity and the absence of the discrete tumor specificity of the fluorophore becomes an issue for the tumor margin, and of course for the image-guided surgery. So following the concept of tumor cell activation of molecular targeted fluorescence probe to provide the mechanism of delineating tumor margins, in a very preliminary xenograph study, we simulated the fluorescence image guided surgery by the co-administration of EGFR specific. EGFR is epidermal growth factor receptor, which is a cell surface receptor and overexpressed in various cancer cells. So we co-administered an EGFR-specific antibody cetuximab and simultaneously a non-specific IgG reference intravenously into orthotopic pancreatic cancer xenograft and acquired the hyperspectral NIR fluorescence images. So here in this image, we can see that the tumor margin is clearly visualized within the pancreas by the cetuximab-based targeted probe, whereas the reference IgG probe is showing non-specific accumulation with the significant background in the pancreas and the surrounding organs. And further, when we generated the heat map image using the FIRE lookup table, it is quantitatively showing the specific binding where the set is, this yellow is the high and the scale, there is a scale from low to high. So the specific binding where the set is above the IgG non-specific level. Thus, it is delineating the tumor margin more accurately when compared to the activatable set alone. So this combination of molecular targeting with the dual probe imaging circumvents the presence of extracellular non-specific signals that we were able to see here in the case of non-specific and providing the enhanced contrast. Furthermore, this multicolor activatable imaging platform have the potential to be further developed as theranostic, because that theranostic platform would have, in addition to molecular targeted fluorescence, also have a therapeutic agent that can add cytotoxic reactive species, and the problem can solve the problem of the residual tumors, because it will enable the surgeons to specifically, on the first place, specifically outline the tumor, and then on the second place, selectively mop up the residual microscopic disease. And that can further improve the chances, reduce the chances for the recurrences. So these ligand-targeted nanoliposomes, which are emerging nowadays in the clinical trial for the numerous cancer indications, and could be an ideal candidate for a theranostic platform because of their ability of carrying high payloads in their different compartments based on their hydrophobicity or the hydrophilicity of the molecule. Moreover, their surfaces can be chemically tuned and modified for the multiple ligands binding. So they not only enhance the localized delivery of the multiple therapeutics, but also decreases the systemic toxicity. However, validating receptor engagement and cellular targeted delivery of that sort of nanotherapeutics in vivo especially is a complex challenge. And because that is partially skewed by the dominant EPR effect, as I mentioned previously, and that because this also only inform about the delivery to the tumor, resulting in the tumor to normal matrix. So to this end, for the true tumor receptor engagement of the targeted nanomedicine, and thus to quantify the receptor-specific binding capacity of the ligand, especially in vivo, we have developed an NIR-activatable targeted platform that is consisting a nanoliposomal core that is covalently coupled to a fluorophore, which is 680 here, iodide 680 here, and to EGFR targeted ligand, a monoclonal antibody cetuximab through the copper-free click chemistry. And in order to be able to quantify the in vivo concentration of this targeted nanomedicine, that binds specifically to, we also developed and optimal reference contrast to account for the passive accumulation due to the EPR effect. So this reference contrast, which is conjugated to the irrelevant IgG molecule, is completely matched with this targeted nanomedicine in terms of size, its zeta potential, that's the charge, and the antibody surface density on its surface, and the optimal brightness. These are the basic characterization when you deal with the nanotherapeutics. So for the non-invasive quantitation of our targeted nanomedicine binding, we co-administered the TNM and the reference intravenously to the mice which are bearing human tumor xenografts. And here are the representative images of 3 tumors with the differential EGFR and differential EGFR expression is with the 9L being the null for this receptor and U251 being the high. And these are all 3 brain glioma cell lines. So here the green images that are shown green here correspond to the non-specific reference uptake, and the red are showing the specific contrast with the targeted nanomedicine alone and the delivery of TNM to the tumor. So using these quantitative molecular images and the uptake of the untargeted reporter as a reference tissue, we derived the single time point binding potential values, which is a unitless measure of the product of binding affinity of a ligand, which is cetuximab here in this study, and the receptor concentration, and the receptor under consideration here is the EGFR. But before moving to further quantitation, we first validated the tumor delivery of the targeted nanomedicine using photoacoustic imaging. And as we have used the visual sonic Vevo system previously in one of our study that was purely therapeutic based. So, and in that study, we have used the system for the tumor penetration, and where the tumor were imaged to generate 2-dimensional mapping of the tumor distribution of TNM, and the validity of these photoacoustic signals that are coming from the IRDI 800 that we used as a photoacoustic contrast agent before and after the administration. So using randomly selected vessels from this 2D cross-sectional images from the mice, TNM penetration was visualized, and that was really helpful for the effective therapy. So the same for this study, we did that because as wild field fluorescence imaging is restructured to the surface of the tumor. So we performed photoacoustic imaging in order to establish whether the higher tumor delivery of targeted nanomedicine is consistent throughout the entire tumor depth. So when matched with the anatomical information that's provided by the ultrasound image, multi-wavelength photoacoustic imaging shows that our targeted nanomedicines that are shown pink here is in fact distributed throughout the entire depth of the tumor when visualized after 24 hours of the induction, and present a higher amount than the sham to all the tumor Z-planes. And the green arrows here are showing the low accumulation of the non-specific probe. So this was the validation of the tumor TNM delivery to the tumor as we did with the fluorescence images and the photoacoustic one. So quantifying the in vivo molecular specificity of the targeted nanomedicines, again, we calculated the binding potential measurements that were derived from the molecular images. We back calculated the in vivo concentration of the receptor EGFR in the three-dimensional space. And this EGFR density or EGFR concentration that is plotted here at the y-axis is taken as a direct measure of molecular specificity. And the x-axis is showing the time, different time points, 24 hour, 48 and 72, and in different tumors. So the TNM was, as we can see from the plot, are shown highest specificity in U251 tumors, that is about 2.15 nanomolar, followed by U87 tumors, then the lowest for the 9L tumors. This value is closely matching with the experimental in vivo EGFR concentration that is expressed by U251 tumors, bioavailable for the receptor. And a statistically significant correlation was also found between the EGFR concentration that the TNM bound to in vivo that are calculated here, and the EGFR receptors per cell in vitro that were quantified using the flow cytometry. So this quantitative confirmation is for the in vivo specificity for the EGFR for 3 tumors with the differential EGFR expressions. And then quantitative, for the quantitative EGFR specific contrast for the image guided surgery, as was the goal, we generated binding potential images of the three tumors, whereby the contrast that can be shown here in these images is provided a direct this contrast is directly proportional to the EGFR concentration that we have back calculated and plotted in the previous plots. So these are the representative images, and here it is easier to locate the tumor based on this contrast than with the targeted or the non-specific fluorescence imaging alone that we have shown previously, and it's best described in the comparison of the split images. That the imaging of a single targeted nanoconstruct alone is not reliable for detecting the specific binding or visualizing the tumor based on their expression status. So a non-specific reference is always essential if you want to quantitate the image and the true specificity of any targeted nanomedicine for the receptor. So importance of generating these binding potential images with the EGFR concentration specific contrast is that the image intensities are not dependent on the nanotracer signal strength, but are internally normalized, and the information and informing the degree of the EGFR for the application in image guided surgery or image guided therapy. And we further did some quantitation for the molecular specificity and characterized the optically derived area under the curve for both targeted nanomedicine, that is calculated as the average over 72 hours period time. And this plot is showing the mean value for the reference, represent the non-specific tumor accumulation, as we mentioned that through the EPR effect. So these profiles are following the trend that is representative of the receptor expression level in 3 types of the tumor, U251 being the highest and 9L being null or the lowest. Further optical pharmacokinetic analysis, which are not shown here, also provide the information that longitudinal tumor exposure can be increased up to 8-fold with the targeting. So this first demonstration of non-invasive quantitation of targeted nanomedicine binding and in vivo molecular specificity here, introducing a previously unexplored approach and concluding that the ligand-targeted nanomedicines are well capable to quantify the true molecular specificity in vivo. And moreover, the misinterpretation of tumor to normal matrix can be avoided by utilizing this approach. This concept can be expanded to quantify molecular specificity of any nanotherapeutics, targeted nanotherapeutics, or for any receptor targeted in the tumor except EGFR. We just use EGFR here, but that can be applied to any tumor type that overexpress a receptor, given that the predominant in vivo expression level exists there, or can be derived from optical biopsies using an appropriate benchmark. So this non-invasive paired nanotracer imaging approach can improve the sensitivity and the specificity over the conventional fluorescence imaging application that we show in the first slide, and specifically help in fluorescence-guided surgery and directly approximate the receptor status of the tumor. And that could be valuable information for the choice of the therapies after the surgeries. As we said previously, that the initial multicolor platform can be developed into a theranostic one. So we are further exploring this by encapsulating the multiple treatment modalities that exhibit non-overlapping modes of cytotoxicity for the complex heterogeneous in vivo tumors. And so that we can further mitigate the risk of the treatment escape. And most importantly, the two challenges that were mentioned initially, that the receptor-specific contrast for the selective image-guided surgery, and after the surgery, the specific eradication of the residual microscopic disease that left after and causes the recurrence or metastasis. So with this, I would like to say thanks to my mentor, Professor Taiba Hassan, for being a great mentor and being my inspiration in the field of photonanomedicine and shaping up my scientific career. And of course, the NIH funding, without which no research is possible. And I would like to say thanks specifically to my lab fellows for their continuous help and their support throughout the project in specific and in general. And to Dr. Jirjis Abed for leading the project, and our collaborator Kimberly Samko from Thayer School of Engineering, and Dr. Malidi for providing her expertise for the photoacoustic imaging and the image analysis when she was here at the Wellman Center for the Photomedicine. And thanks again for the visual Sonics for sponsoring my AACR conference and providing me an opportunity to talk to you guys and share our exciting work with you guys. Thank you so much.
Thank you so much, Shazia, for that really exciting and interesting presentation. At this point, I want to encourage everyone to use that Q&A button on the bottom of your Zoom panel to share any questions you might have. I just wanted to share with the group one final thing before we move over for Shazia to respond to your questions. And that's that we have an upcoming virtual workshop that I wanted everyone to make sure they save the date for. So the date is October 6th, 10 a.m. We'll be hosting a virtual molecular imaging workshop. And this is where we will discuss various uses of the Vevo technology for molecular imaging. So some of the things we could be discussing or showing is the use of targeted photoacoustic imaging for, as you can see in the top left here, in this sub-Q tumor, we can see a nanoparticle targeting within that tumor. We can also do molecular imaging using ultrasound and microbubble contrast. So in this example on the top right, we see tumors and differential expression of VEGF through targeting of those contrast microbubbles, and we see differential expression within that tumor. We can also look at whole body biodistribution of targeted agents. In this case, on the bottom left, we have angiostamp, and we see its accumulation in the kidneys, and we have a more broad whole body view of the mouse in this case. And the angiostamp in green is indicated here, and those are the kidneys. And then another topic of discussion is targeted drug delivery. So in this image, we can see drug accumulation as seen photoacoustically and accumulating over time within this orthotopic colon cancer model. So these are just some examples of how Vevo technology can assist in molecular imaging, as Shazia alluded to in her presentation. And we'll have some really nice speakers and live imaging sessions. And we'll be coming out with an agenda shortly, but please stay tuned to our website or contact us, and we'll make sure you get the invitation and are able to register for this exciting event. So at this point, I would like to open the floor for any questions you might have for Shazia. And I see there's a couple trickling in right now. So someone had commented, Shazia, great talk. Why the choice of ligand? Do you have specific clinical applications in mind?
Yeah, it's a great question, and I think I when I started my talk, I made it very clear with the example that the first thing is this the choice of the ligand or the targeted either it's diagnostic or therapeutic. The reason is this with the untargeted probes, untargeted nanotherapeutics. The major problem is this: it is taken up by the EPR effect and goes everywhere in the body and destroy or can cause damage to the healthy tissues as well. So as the cancer tissues, cancer cells, they specifically overexpress different cell surface markers, and each this epidermal growth factor receptor is one of them. So utilizing this presence and these markers, we can plan and design the therapies or the nanomedicines for the specific imaging or for the specific or the targeted therapies for the cancer, so that avoiding the healthy tissues. That's the one answer. And then for this specific EGFR, as I said, that is because it is overexpressive in almost all of the type of the cancers, and its overexpression is relatively very high than the healthy tissues. So targeting using this as for the targeted approach is always beneficial, and as I have shown in this, the non-specific and the specific uptake for the tumor delivery of the targeted nanomedicines. I hope I have answered your question.
Yeah, that's great. Thank you. And next question is more generally about the fluorescent guided surgery approach. Is there certain types of cancers that this is more applicable for? And similarly, a follow-up to that, for the EGFR specific contrast that you've presented here, are certain cancer types or locations going to be more applicable?
Yeah, this is a very, very good question because, of course, fluorescence image guided surgery is a very exceptional or a very good tool for the tumor which are localized. And for the localized tumor, no matter which X receptor they have, so the fluorescence image guided surgery or just imaging work. But the real problem comes when you are dealing with a tumor that is disseminated, like the ovarian cancer. It's all over the peritoneal cavity. So there is a specific need for a targeted or a specific version of your imaging or a therapeutic agent, so that all parts or the solid tumor chunks that are all over the peritoneal cavity can be seen. And this fluorescence image guided surgery with the specific contrast agent or with the targeted contrast agent can be helpful in every type of the tumor. It's more for the disseminated tumors.
Perfect, thank you. And in the interest of time, we'll do one final question here. And if anyone does come up with any additional questions, please contact us and we can pass those along to Shazia. So the question comes with a compliment at the start. Thanks for the nice talk. I want to ask whether you could also monitor the release of drugs over time using the photoacoustic system after post-administration, or any comments you have regarding the control release and what was the minimum concentration required to get a photoacoustic signal.
Yeah, that's a great question, and I think it's an obvious question to relate this with the talk, because as I said that this is the first demonstration and it's kind of a platform that's been developed. No matter which imaging technique is used here, and of course there are some limitation of the fluorescence imaging as well while doing the quantitation. So that's why we used photoacoustic imaging for the validation, and specifically in our previous study when we used it for the targeted nanomedicine penetration through the blood vessels. So doing different time points photoacoustic imaging can give a better idea of its biodistribution profile before the surgery, before the any treatment, either it's a imaging or any type of the therapy, and after the therapy, it can also help in monitor for the clearance of the therapeutics, because that's the clearance from different parts of the body is also an important information. So I think, and another comment I want to say here, that the platform is so flexible that this in vivo receptor quantitation can further be explored by using photoacoustic imaging. Yeah, in any type of orthotopic or subcutaneous tumor model.
Perfect. Thank you so much, Shazia. Again, in the interest of time, if you have any further questions, please feel free to contact us and we can set you up with Shazia to address those questions. Otherwise, thank you so much, Shazia, for your excellent presentation. Thank you for everyone for joining us today. Please visit our website and contact us using our web forms on our website if you have any questions. Please continue to visit our webinar page in case you want to register for the next webinar or view our past recordings. You can find us on all social media channels. And please visit our conference page on our website to view any virtual booths and virtual conferences we'll be attending in the future. Also, I want to point out that we do have a training portal available to you, the Learning Hub. So if you want to view any training videos, please go on our website and search for the Learning Hub. Thank you so much, and I hope everyone has an excellent day.